All right, and just to give you a little bit of background on Shweta, so she's a fourth year PhD candidate in the Department of Medicine here in the Division of Infectious Diseases, and she's working under the supervision of Dr. Joel Dax, one of the organizers and active member of our organizing committee. The goal of her PhD thesis is to understand the evolution and mechanisms of critical cargo transport protein machinery in the enteric microbial parasite, Giardia, which is responsible for the diarrheal disease Giardiasis. She's interested specifically in studying the cargo transport machinery and Giardia because it plays crucial roles in this pathogen to transition between various stages of its life cycle, evade host immune responses, and establish infection in both humans and animals. To comprehensively address questions, she's using a systems approach by combining evolutionary bioinformatics, population genomics, and molecular parasitology methods. The interdisciplinary work is being performed with at least 3 different research groups, that is internationals, the University of Zurich and the University of Bern in Switzerland, and it's also cross-institutional within Canada with Dalhousie University in Nova Scotia being collaborators. Thank you, Shweta.
All right, let me just quickly share my screen. Perfect. Is everybody able to see it and hear me?
Yes, thank you.
Thank you for the introduction, and I apologize for a bit of a change in the title. I'm going to be providing a brief overview of my research and what I've been doing as part of my PhD, and switching directions a little bit from the talks that we've heard so far. So I'm going to be providing some more molecular, basic cell biology type research, which I hope will be interesting to the audience. So yeah, so my name is Shweta, and I'm a fourth year PhD student in the Division of Infectious Diseases. And today I will be talking to you about various evolutionary and functional dynamics of the membrane trafficking system in the gut parasite Giardia. And so just to provide some more context and some background on Giardia, Giardia is a microbial eukaryotic parasite that causes the disease giardiasis, which is also known as beaver fever, a term that you may have heard of. It's prevalent in North America, including in Canada, and it is globally prevalent and particularly higher in resource-poor settings. And so it is a food and waterborne diarrheal disease that causes a significant burden on the healthcare systems around the world. It is especially problematic, and cases are often seen in younger children, and there are often outbreaks reported in daycare centers. But apart from kids, the World Health Organization estimates about 300 million individuals to be infected each year. And so in this map, I hope to have demonstrated that Giardia is present and affects virtually every single population and countries across in various continents, including in Canada. And so this is what the parasite looks like. It's a single-celled microbial eukaryote. It has 8 flagella, a ventral disc that attaches to the gut wall. And this is kind of how it establishes infection once it enters the host. And so in terms of causing the actual infection, aside from humans, Giardia can infect other animals. So it has a anthropootic and zoonotic potential. And in fact, in humans and animals, Giardiasis is specifically caused by the species Giardia intestinalis or Giardia lamblia. So those are 2 interchangeable terms that you may have heard of. But human Giardiasis is specifically caused by assemblage A and B, which has the greatest host range and zoonotic and anthropootic potential. And so Giardia is an enteric microbial eukaryote, and it's a parasite. So its life cycle and the disease establishment involves the animal gut. It is a simple life cycle, unlike malaria, which was mentioned previously, in that it consists of an environmentally resistant cyst stage, which are ingested through food or water. These enter the human gastrointestinal tract, and here the pH changes as well as secretion of parasite-specific factors trigger a transition to feeding forms known as the trophozoites, and these are the ones that cause the actual disease in the host. Here they replicate, they acquire nutrients, they release pathogenicity factors to evade the host immune cells, and are also functioning to disrupt the gut-brush border, which causes the diarrheal symptoms. And so as the parasites move along the small intestinal tract, changes to an alkaline pH and bile secretions trigger the pathogen to secrete cyst wall proteins, which allow them to transform back to the cyst stage, which are the environmentally resistant stages. And these are excreted into the environment where they can remain dormant for months. And so throughout the life cycle, Giardia is constantly secreting parasite factors and uptaking host nutrients. And so for this, it heavily relies on something known as the membrane trafficking system. So just very briefly, the membrane trafficking system is a hallmark feature that's present in all eukaryotic cells, including humans, plants, yeast, anything that is a eukaryotic cell. And the membrane trafficking system is a network of organelles and protein machinery that include things such as the endoplasmic reticulum, the Golgi, the endosomes, lysosomes, as well as numerous cargo and protein machineries that coordinate trafficking of cargo internally within the cell and also in and out of the cell. But when we look at Giardia's endomembrane organization under the microscope, it looks to be quite different compared to what you would expect from a normal eukaryote. And so it seems to be missing key organelles like the pancake-stacked Golgi, various different vesicular compartments such as the endosomes are also missing. And instead, it has these unique Giardia-specific compartments that are specifically present in this parasite and are not seen anywhere else. And their functions and evolutionary histories are still very poorly understood. And so in order for us to understand how it causes disease by secreting those factors, those parasite-specific virulence proteins into the environment, into its host cell environment, we need to understand how this machinery function, how this system functions, but also how it has evolved over its evolutionary course. And so because it's using this system to cause the infection and evade the host immune cells, there's interest in understanding, what it even is. And so these Giardia-specific compartments include these really unique organelles that are known as these peripheral vacuoles. And these are vesicle-like structures that are present ubiquitously close to the cell periphery. And so work by us and others in the field have postulated that these are likely a major interface for cargo sorting and secretion and may represent potentially fused forms of the missing endosomal compartments, although there's still quite a lot that we don't know. Next, it has a very expanded endoplasmic reticulum that often makes contacts with these PV organelles and are also thought to be endosomal in nature. They're very dynamic and are thought to be highly dynamic in their function, in that they're capable of doing many things such as protein synthesis, cargo sorting, and processing of cargo that has entered the cell. So again, it's quite an enigmatic organelle, which is not very well understood. But the best understood compartment are the state-specific encystation vesicles that are thought to be performing the Golgi-like functions. And so these vesicles only emerge prior to encystation to carry cyst wall proteins that decorate the parasite surface, which allow the feeding stage, the trophozoite stage, to transition to the environmentally resistant cyst stage, and provides for a protective barrier onto the pathogen to be able to survive. And so I hope to have demonstrated that the endomembrane system in this parasite is quite bizarre. And so for us to understand how it got to the way that it did, we need to take cues from its close free-living and parasitic relatives. And so under the microscope, Giardia's free-living and parasitic relatives have a varying degree of complexity in their endomembrane organization in comparison to, again, model eukaryotic cells that have been studied like yeast or plants or animals. And so what we hypothesize is that this organization and functional complexity within the cargo trafficking system in Giardia has been streamlined as a byproduct of gradual evolutionary modifications, some which predate Giardia, but several that are a byproduct of a specific adaptation to the host. And so by studying proteins that govern functions and biogenesis of let's say, typical trafficking organelles in model eukaryotes and seeing how they have evolved and how they function in Giardia, we can start to gain insight into how this process has occurred over time. And so for this, I am undertaking a bioinformatic approach, but also some molecular investigation into the evolution, function, and the homology of the membrane trafficking system in this pathogen in Giardia as a whole, but also to examine for potential strain-specific differences in the Giardia intestinalis lineage. And so we've examined several molecular systems. However, I will only give a snapshot of just a one Dominique has mentioned, which is the ARF GTPase regulatory system. And this consists of several protein machineries that function together to act as molecular switches within the cell for cargo vesicle trafficking of material between the Golgi and for post-Golgi trafficking. And so using a combination of comparative genomics and phylogenetics, quite a neat story in that the system is quite stripped down from the very beginning in this lineage, in that a lot of the losses have occurred prior to the Giardia lineage, but also in the free living relatives. But interestingly, there were also Giardia-specific losses and duplications, specifically in the human infecting and the zoonotic assemblages. And so now to follow up, we wanted to look at some of the molecular functions of these proteins in vitro and to see what they're doing in the cells of these parasites, which is a project that I'm currently working on in collaboration with the group at the University of Bern. So this is just an example of some of the data that I've collected so far. Some of the proteins to have functions at those peripheral vacuoles, so the cell periphery, where they might be involved in cargo uptake. And now we're testing their additional localizations and molecular interactions. If we lock them in specific on and off stages, and what machinery do they interact with, and if we introduce specific types of mutation, how does that affect their localization within the cell? And so for this, we're doing some immunofluorescence microscopy to look for whether there are changes in localization, as well as immunoprecipitation and mass spectrometry to how this interaction, this dynamic changes. So aside from general streamlining in Giardia lineage as a whole, like I showed, our previous comparative genomic work, not only in this system, but many other protein trafficking machineries that I've investigated, we've seen that there are considerable genetic and protein level differences. And work by other groups in the field who have done a lot of genome sequencing of Giardia, specifically of the human infecting assemblage in B isolates, have also prompted many to suggest that intestinalis is actually a species complex, and that in the humans, this is potentially caused by two different species. And so this is these underlying biological differences might contribute to differences in disease severity or symptomatic versus asymptomatic presentation. So giardiasis can actually manifest as a completely asymptomatic disease, let's say, in healthy individuals, but can cause symptomatic disease also in specific types of populations. And so it's important to investigate to what degree and to what extent these differences are occurring. And so again, to do this, we need to do more isolates at a larger population genomics level. And so previously, the British Columbia Center for Disease Control in Canada had archived about 90 isolates of assemblage A and B cysts from surface waters and beavers to assess for some of the epidemiological and transmission factors that could have played a role in the provincial outbreak that had occurred in the late 1980s. And so these were recently subject to being resequenced using whole genome sequencing. And so now I'm just undertaking a large-scale assembly of these isolates to investigate some of the questions pertaining to these genetic and cell biological differences, including in the tracking system proteins, but also in some key virulence genes that may have role in causing disease and disease severity. And so I hope to present those results in a future global health round and a future conference. I'm sure that will be of interest, especially from a public health standpoint, and to even shed light as to why giardia sometimes causes symptomatic disease versus not. And so with that, I will conclude my presentation and thank members of the DAX lab, as well as both of my supervisors, Dr. Dax and Dr. Croxon, as well as various collaborators who have made interdisciplinary work possible. Of course, funding agencies that have supported both the computational and the bench research aspects of this project. And thank you for inviting me today. I look forward to hearing all of the other presentations later. And yeah, thank you all for this.